Now we are looking at transgenic crops. What are the beneficial effects of transgenic crops in Indian agriculture? When you take transgenic crops are genetically modified. They are genetically modified, and we have several applications of transgenic crops. A very important application of transgenic crop is you can develop transgenic tomato. Now, transgenic tomato is popularly commercially called as flavor saver tomato. The flavor saver tomato is a tomato which is developed by genetic engineering, and it prevents softening of the sort of a tomato. So, under extreme cold conditions, the tomato does not soft, or it does not lead to rot of the tomato. Thereby, it increases the shelf value of tomato or the keeping quality of tomato. Thereby, in transit, tomatoes can stay for a very long time, and it has got a lot of commercial applications with relation to development of tomato ketchups, sauces. The second important application with relation to transgenic crops relates to rice. In rice, we have one variety of rice called the golden rice, which has been developed by Potricus and Bayer. And transgenic rice has been one success experiment with relation to transgenics. So, in the case of rice, the golden rice is fortified, is biofortified, that is, it is enriched with vitamin A. Because rice is enriched with vitamin A, it plays a very important role with relation to preventing blindness in poor, underdeveloped countries, especially with relation to children. So, children who are fed with transgenic rice have the sort of a rice which is biofortified or which is enriched with vitamin A, which can prevent blindness. Thereby, we understand here we have a sort of an application wherein we take biotechnology, which has got a sort of a long-term effect. For long term, for several generations, people would primarily, whoever are consuming transgenic rice, would never have the sort of a situation of blindness. Then third, we will also be learning about Bt cotton, which in Bt cotton we have got the Bacillus thuringiensis. The Bacillus thuringiensis spore produces the toxin, an endotoxin, which when consumed by the insect in its epithelial gut in the presence of an alkaline pH becomes toxic, and it can lead to killing of the sort of an insect. Thereby, we have the development of Bt cotton as well as Bt brinjal, which are all important sources of biopesticide, a microorganism which can function as a sort of a biopesticide for the control of very important pests in cotton and brinjal. Then we come to brassica, that is all the sort of the mustard family. In the mustard family, we can develop transgenic brassicas. Now, transgenic brassicas are transgenic mustard plants into which we can put the sort of a gene which synthesizes hirudin. Hirudin is a very important blood clot factor. So, thereby we can exactly transfer the hirudin gene through the sort of brassicas. Next, we come to herbicide resistant crops. When you take the herbicide resistant crops, the most popular of the herbicide resistant crops are the maize and soybean. So, in maize and soybean, we have developed primarily transgenic crops which are termed as Roundup Ready, depending upon the name of the company. It is called Roundup Ready, and this is the commercial name. And Roundup Readies are varieties of maize and soybean which have been developed by transgenic technology, and they are herbicide resistant. Next, we have the wheat plant. Wheat plant, that is Zea maize, is also developed for resistance to pesticides, and we have a local variety which is called Besta. Besta is nothing but an application of wheat which is transgenic to pesticides. Potato, Solanum tuberosum, is improved by transgenics, by biotechnology. It has been improved for higher starch content. Tobacco is improved for nematode resistance, which is popular in the roots. We will be learning more about tobacco as well as even golden rice and also about flavor saver tomato at a later period. Then we have basmati rice. Basmati rice is a transgenic rice which has been developed for biotic as well as abiotic resistance. And we have several sort of fruit strains, among them being papaya. Papaya is resistant to viral resistance. So, when you take papaya, papaya is resistant to ring spot virus. It is resistant to ring spot virus. So, transgenic papaya are developed so that it becomes more resistant to ring spot virus. And similarly, if you take cotton, we understand cotton primarily it is resistant to insects. It is resistant to insects. And tomato primarily it is resistant again to a bacterial pathogen which is termed as Pseudomonas. Tomato is resistant to Pseudomonas, which is a sort of a bacterial pathogen. Then again potato is resistant to a very important, we can breed potato which is very important for a pathogen which is termed as Phytophthora. Phytophthora infestans has led to a disease which is termed as late blight of potato, which has led to a lot of epidemics and the migration of the Europeans to America. So, here if you want to breed potato for resistance to Phytophthora as well as tomato to Pseudomonas, which is a very famous sort of a pathogen of tomato, we can develop them by means of transgenic engineering. Now, similarly when you take food processing, as I told you in the case of tomato, it becomes resistant, flavor saver tomato gets resistant to primarily for against softening, and thereby you can improve a sort of a tomato for primarily for making it more hard. It's so that you can improve its sort of a storage value, you can enable it for better transport, and it is primarily for delayed ripening. It is for delayed ripening and long shelf value. And similarly when you take rice, rice is improved for higher nutrition quality. The nutrition quality in the case of rice is very important. It is enriched with vitamin A to prevent blindness. And even in the case of brassicas, we can develop male sterile lines. We can develop MSL lines or male sterile lines. Now, male sterile lines primarily enable the sort of a male pollen to be sterile, so that we can easily pollinate the entire sort of a crops by an aerial spray of pollens. So, thereby you can prevent the problem of manual emasculation, and it helps in improving the sort of a it helps in getting down the sort of a cost of the hybrid seed. So, male sterile lines enable prevent manual pollination, and we can do artificial aerial pollination. And similarly when you take basmati rice, it is as I understood you can easily break it for both abiotic and biotic stress. Now, when you take transgenics, you understand if you take papaya is resistant to ring spot virus. Tomato for Pseudomonas. Pseudomonas is a very important sort of a wilt disease. And if you take potato, that is against the sort of in potato it is against the sort of a late blight disease. And so we understand when you take transgenics, you can develop transgenics for resistance to both pathogens and pests. And the transgenics also have applications in food processing. It has got a very important application with relation to food processing technology. It can also improve the nutritive value in the case of golden rice, and it is also important for hybrid seed production, as we have seen in the case of Brassica campestris. And transgenics also have applications with relation to biotic and abiotic stress, as seen in the case of primarily in the case of basmati rice. So, the potential applications of transgenics are with relation to improving nutrition, keeping quality means frost resistant, as in the case of tomato, and resistance to bollworm infection, tolerance to herbicide, and also being high yielding. And we understand it plays a very important role with relation to prevention of night blindness. Now, what exactly are the benefits and risks of transgenic crops? When you take transgenic crops, all transgenic crops always it helps the benefits of transgenic crops. Now, we would be making a comparison between transgenic crops. What are the benefits and risks of transgenic crops? if you take the benefits and if you take the risks, or what are the sort of limitations? The first important benefit or crop, we have increased crop productivity. By transgenic crops, you can definitely cause an improvement of the yield, because we have a high yielding gene which can primarily enable the plants to grow and adapt to adverse environmental conditions. It can lead to better crop production, as implied, for resistance to pesticides and also resistance to diseases. For resistance to pests and diseases, it can offer crop protection. Third, it can improve the nutritive value, like as we had discussed with relation to rice. Nutritive value with relation to rice. It can lead to better flavor, as seen in the case of the flavor saver tomato. The keeping quality of the vegetable can be improved upon, as seen again in the case of frost resistant or maybe bruise resistant tomato. In the flavor saver tomato is also bruise resistant. That is a prevention of any sort of an injury. You can get fresher produce which can remain year long. There are environmental benefits, because it does not have the application of a sort of a pesticide or a sort of a fungicide, and again they are tolerant to abiotic stress. So, when we address to the benefits, we have 8 benefits: crop productivity, high crop production, high nutritive value, flavor, keeping quality, fresher produce, environment benefits and tolerance to abiotic stress. Now, let us look at the limitations of the risks. Whenever a new technology comes in, there is always the plus and minus of the sort of a technology. But however, in many situations the benefits are greater than the risks, and even when there is a sort of a risk, if there is a problem, there will always be solutions to the sort of a problem. That is how primary sciences evolved. So, in the perspective of good biotechnology, good biotechnology has got lot of applications in the 21st century. Now, the possible risks or the limitations of biotechnology is it can lead to the development of allergens. This is the common sort of a fear among humans. So, its social or ethical acceptance is questionable, because it may lead to lot of allergens. There could be a buildup of toxicity. It is believed that when we have transgenic crops, the toxicity buildup within the sort of a plants could be very, very high. And there is lowered antibiotic resistance by the crop, because as we understand, when you do transgenics, there is high possibility that even the sort of microbes which cause a disease can easily get mutated and they can change. So, it can lead to lowered antibiotic resistance. There is a fear of gene escape. A transgenic gene, supposing we are planning a transgenic, planning to develop a transgenic crop while we are doing the breeding, and supposing your pollen carries the sort of a foreign gene of interest. If it is let loose, if the pollens are let loose in a sort of a field in which there are more of weeds or more crops, there is a fear that there could be a sort of a gene escape. And it is sure that whenever we have transgenics, a transgenic weed, when it is developed, it can develop into a sort of a super weed which is larger in size, larger in biomass, and it can compete with the local crop, and we could have the more of weeds and less of crops. And again, the people are skeptic whether there can be a sort of an insecticidal resistance break, because after a particular period of time, definitely the insects as well as the pathogens, because they are primarily microorganisms, and microorganisms sporulate at a very fast speed and they become more resistant to any of these sort of factors. There is a fear that when you go for transgenics, there is a loss of biological diversity, there is variety and variability of life forms, and the social and ethical acceptance. So, this way, when we understand, when you see transgenics have got both the benefits as well as risks as two sides of the same coin, and the benefits primarily seem to outweigh the sort of risks in the 21st century, with the advancement of molecular biology, with the advancement of bioinformatics, and with the advancement of technology which integrates, like if you take a sort of a nanotechnology. If you take nanotechnology primarily and biotechnology, when they are fused together along with good science, a predictable science, and also a sort of a very intense deep science, it can lead to several of the sort of a solutions to the present day problems in agriculture. Now next we would be primarily touching upon with relation to transgenic rice, and in transgenic rice, as I told you, we come across a very important word which is termed as biofortification. Now what exactly is the meaning of biofortification? We are finished on the risks and benefits and risks. We are about to enter into the entire study of transgenic rice, or the form how golden rice has been a sort of a success story, or maybe the golden rice is just like the sort of a Trojan horse. Like many people have awaited what is the sort of a success or the failure of biotechnology in terms of the transgenic rice. Now biofortification is a method wherein primarily it is one component of plant breeding, and in biofortification we aim at improving the nutritive quality of a crop variety, the total nutritive quality of a crop variety. And if a particular crop is enriched with vitamins and minerals or higher proteins and also healthier fats, and the most important objective of a biofortified food is to improve the health of people. Now when you take transgenic rice, transgenic rice, it has been genetically engineered. Transgenic rice has been genetically engineered by two important scientists called Potricus and Bayer from the Swiss Institute of Zurich. Primarily what they had done is, they had found out that when you take rice has got an embryo and an endosperm, and in the endosperm there is starch synthesis. So they had manipulated, or by genetic engineering they had taken two genes from bacteria and one gene from daffodil, and they had primarily synthesized beta carotene. So when they synthesized beta carotene in the endosperm of rice, we could get transgenic rice, and the transgenic rice had the sort of a precursor for vitamin A. The details of how the transgenic rice has been developed is now being illustrated. Now specifically in biotechnology and its applications in terms of food production, we are going to study the story of transgenic rice. Now transgenic rice is genetically modified rice or genetically engineered rice, and it applies the principle of recombinant DNA technology. When you take transgenic rice has been evolved by Peter Bayer and Potricus from the Zurich University at Switzerland. Now what are the objectives of transgenic rice? It was believed that if rice is fortified with vitamin A, it has got fortified with vitamin A, it can lead to prevention of vitamin A deficiency in children, that is blindness in children. So keeping in mind that biotechnology is aimed at improving the quality of human life, biotechnology is aimed at improving the food quality, both Potricus and Bayer have developed a very important sort of a transgenic rice to prevent blindness, preferably prevent blindness in children. Now when you take rice, the transgenic rice has evolved into a very popular or a commercial name which is termed as the golden rice. So what happens in the golden rice? There is biofortification. It is enriched, there is enrichment with nutrients. Which is the most important nutrient? Vitamin A is the most important nutrient. So how do you get vitamin A? You get vitamin A when you synthesize, it is synthesized from beta carotene. Now beta carotene is a precursor of vitamin A. Now understanding that in rice we have starch synthesis, and starch synthesis occurs in the endosperm, both Potricus and Peter Bayer tried to genetically engineer the rice. So they had the sort of a plan that when you take a normal rice plant, in a normal rice plant, you have a sort of a precursor called geranyl diphosphate. Geranyl diphosphate is a chemical which is a precursor which is present in the rice endosperm. Given geranyl GGD or geranyl diphosphate, it has evolved from, or it has come from, a chemical compound called IPP. So they found out that rice lacks three very important sort of enzymes. If you want to synthesize or if you want to get beta carotene, you require three very important enzymes. So they had gone on a sort of a search for where the enzymes are present, because if you take each particular enzyme, understand, a gene determines a biochemical pathway. A DNA makes RNA, RNA makes proteins, proteins is equal to enzymes. So thereby you have the gene which can make the sort of an enzyme, and rice by nature lacks the three enzymes. So if ever we could get in the sort of a gene and enable the sort of a gene to express in the endosperm, coding for the sort of an enzyme, then maybe we could reconstruct the sort of a pathway. So they were in the process of adding three genes to the sort of a pathway. So their transgenic rice is produced from three important pathways with adding three genes, so that the pathway is very much complete. Now we have to start with, we have geranyl diphosphate, which is a precursor. If to rice you add in, by genetic engineering, you add in the first sort of a gene, which is a gene from daffodil. A daffodil is an ornamental plant, it's an ornamental weed, it's called narcissus. So when you take the gene from daffodil, the gene from daffodil is from that you can get an enzyme called phytone synthetase. Now phytone synthetase is abbreviated as PSY. So we have the first important input is from gene 1, you got phytone synthetase. It's very important that you remember the sequence of the genes and also the name of the sort of enzymes. So once there is phytone synthetase, you have the formation of phytone. So geranyl diphosphate, that is GGD, is converted to phytone. Next, they had isolated two important genes from bacteria. From the bacterial gene, from the first gene from bacteria, where they had isolated a gene which codes for phytone desaturase. Now phytone desaturase is abbreviated as CRT. This is also termed as beta carotene or phytone desaturate. Beta carotene desaturate, or it is also termed as phytone desaturate. So in the presence of phytone desaturate, phytone could go to form a sort of a red coloring pigment or a substrate which is termed as lycopene. So lycopene, the production of lycopene, the biosynthetic pathway of the production of lycopene from phytone requires phytone desaturase, which was extracted, which was a product of the second gene or the first bacterial gene in the sort of a vitamin A pathway. Next, from lycopene, they are again taken from daffodil gene, they are taken a gene which codes for lycopene beta cyclase, and it is abbreviated as LCP. So LCY, lycopene beta cyclase, is an enzyme which is coded by the daffodil gene, that is the third gene in the biosynthetic pathway. Now this results in the formation of beta carotene. We understand that beta carotene is a vitamin A precursor. Now when you take the vitamin A precursor, this vitamin A precursor is responsible for the golden rice. It is responsible for golden rice, where three genes are expressed in the rice endosperm. Now when you take beta carotene, to beta carotene, if we add ferritin, what is ferritin? Ferritin is an iron compound. Ferritin is coded by an iron coding gene from beans. If you take beans, Phaseolus has got an iron coding gene, and the iron coding gene is responsible for ferritin. If ferritin is added to beta carotene, there is an overexpression of beta carotene. Because there is an overexpression, that is, a gene get overexpressed, that is, more of the products are getting formed. So an overexpression of beta carotene due to the addition of ferritin results in the color which is termed as yellow or golden color. that is why we have the sort of a name of the genetically modified rice as golden rice. It's called golden yellow rice or golden yellow rice, and primarily it is present in the endosperm of rice. So this has been a very important sort of a success story of transgenic rice. Whenever we take transgenic rice is one of the sort of rices which mainly aims at sustainability and food security. We can get sustainability because in the long run we have biofortified. We have a biofortified rice with a gene the gene for vitamin A is ingrained into the sort of a rice endosperm, and it is a long-term strategy for prevention of blindness, and it relates to food security because we given food. It relates to food security as well as it also relates to the problems of sustainability. Would it be that maybe the transgenic rice is the sort of a first test for biotechnology and genetic engineering, and the partial success, because there had been limitations there, with the sort of a partial success, definitely within a decade or two we could have several sort of applications with relation to biotechnology in the context of food production.